I'm George Carmen. I'm a Board of Governors professor and a distinguished professor of food science. I'm also the founding director of the Rutgers Center for Lipid Research. We study an enzyme known as phosphatidic acid phosphatase. This enzyme catalyzes the penultimate step in the biosynthesis of triglycerides or fat. We know from previous studies that this enzyme is involved in the biosynthesis of fat, and too much of the enzyme can cause obesity, but too little of the enzyme can cause another disease known as lipodystrophy. We found that if we eliminate this enzyme, the lipid precursor that is normally channeled to the biosynthesis of fat is instead channeled to the synthesis of lipids that are deposited in biological membranes. This situation increases the chance of diseases such as cancer and inflammatory diseases and other health problems. Our work on lipid metabolism utilizes baker's yeast as a model system. The advantage of working with baker's yeast is that cells are easy to grow in large quantities, and there's a great deal known about their genetics and molecular biology. This allows us to make mutations almost at will, so we can study the importance of this phosphatidic acid phosphatase enzyme. In this study, we eliminated the PA phosphatase enzyme. And what we found is that instead of making triglycerides in the cell, the cells would make an increased amount of membrane phospholipids. We found that a critical enzyme that's involved in making membrane lipids was activated, causing an increase in an enzyme that initiated membrane phospholipid synthesis. Furthermore, we found that if we made a mutation in the gene for this enzyme, that we could alleviate the increase in membrane phospholipid synthesis, and the cells would actually look normal in spite of the fact that we had also eliminated the PA phosphatase enzyme. The synthesis of fat is important because triglyceride is a storage form of energy. The synthesis of triglycerides are also important because they buffer cells from the toxic effects of free fatty acids. The synthesis of membrane lipids is also important because they serve as structural as well as functional components of the membrane that allows cells to grow. What our studies have shown that the balanced synthesis of triglyceride and membrane lipids is very important, because disturbing the balance can lead to diseases such as cancer or inflammatory responses, or lead to obesity. This implies that if we develop a drug to control the synthesis of fat, we also have to consider the biosynthesis of membrane lipids. So the next step in our research is to understand how to fine-tune the activity of this enzyme. And in this way, we can make sure that cells are not making too much fat, but making enough fat, and also that they don't make too much membrane lipids. The phosphatidic acid phosphatase was actually discovered in 1957 when I was only 7 years old. For a number of years, researchers tried to identify the enzyme, but because of technical problems, it was not accomplished until 1989 by a student in my laboratory named Yiping Lin. From that time onward, people were trying to discover the gene that coded for the enzyme. But again, there were massive technical problems in identifying the gene. It was in 2005 when Gil-Soo Han found a sample of the enzyme that was deposited there in 1993. By that time, the human genome had been sequenced. And through bioinformatics, Gil-Soo Han was able to identify the human gene encoding for the phosphatidic acid phosphatase enzyme. Through a series of experiments, he established the gene-enzyme relationship. And since that time, researchers all over the world have been studying the phosphatidic acid phosphatase enzyme.